thanks a lot for the opportunity. It's really a pleasure to tell you a bit about light sheet microscopy. And later on, I'll give a research talk where I talk a bit more about my research in this area. But I appreciate your time in listening to this, and I know it's not an easy time in India right now. So thanks. All right. So light sheet microscopy is a very diverse topic, and so my intention here is to kind of provide some basics and then provide a couple of specific examples. But really, hopefully, this is just a springboard and launching off point for you to learn more about this technique. So I want to kind of start very generally and just think, how do you optimally acquire an imaging volume? And let's say we take a single cell that you grow on the cover slip. Some of the things you might like to in your microscope images are diffraction limited resolution. You'd like to take that cellular volume with minimum phototoxicity, right? So you don't destroy the thing you're imaging. And then you'd like to image as fast as you need to, certainly fast enough to capture the underlying biology without motion blur. And of course, no fluorescent microscopy technique achieves all of these things all the time, but light sheet microscopy can come close some of the time. So I want to start by kind of just motivating Widefield microscopy. And so in a Widefield or epifluorescence microscope, right, this is kind of a baseline microscope. The defining characteristic is you illuminate the entire sample at once. And I've kind of tried to indicate that here by just this red sort of light coming in and illuminating the entire volume of the sample. And in a wide field microscope, you of course acquire one plane at a time on a camera, but because you're illuminating the entire volume, there's no what we call optical sectioning. So you get lots of background in three-dimensional volumes. And in thicker samples, there can be noticeable photo bleaching and photo damage. But the kind of saving grace of the wide field microscope is that it's very fast, right? You acquire the entire plane at once. And so one way of thinking about this is if you think about what the detector sees, if you have some pixelated camera image, let's say with n by n pixels, here I've shown an example with just 5 by 5 pixels. One way of thinking about the wide field microscope is you have parallelized excitation and parallelized detection. And so because you're illuminating all of the pixels at the same time and you're detecting from all of them at the same time, if you acquire the image with some acquisition time t and you put in some illumination power p, one way of thinking about the wide field microscope is that all of the pixels are giving you information at the same time. And so the signal rate that you get back is just proportional to the exposure time and the amount of illumination power you put in. So in that sense, it's kind of fast. And you can contrast that to a confocal microscope, which is kind of the other extreme, right? So in a confocal microscope, you have a laser beam. Typically, you overfill the back aperture of an objective lens. You form a diffraction limited spot on your sample, and then you scan the sample or the spot relative to the sample while recording point-like on a point detector, like a photomultiplier tube. And the defining characteristic of a confocal microscope is this pinhole, which rejects out of focus light before it's recorded by your detector. The reason the confocal microscope is so useful is that it can provide diffraction limited resolution in samples that are not too aberrating or scattering. You do get optical sectioning because of the pinhole, and it's a workhorse, right? I mean, most everybody has access to a confocal microscope in their university or facility. And it's kind of instructive to contrast the confocal microscope to the wide field microscope, right? So in a confocal microscope, you come in with this kind of hourglass cone of illumination, and the pinhole picks out the region right at the focus. And to build up an image or a volume, you would kind of scan this illumination through the sample, right? Both left and right and up and down and into and out of the page, the slide in this example. And so again, the strength of the confocal microscope is that you get optical sectioning. One of the disadvantages, you get 3D bleaching like a wide field microscope. And one of the issues with a confocal microscope is that it's slower fundamentally than a wide field microscope for the same signal to noise ratio. And when I was learning about confocal microscopes, I thought, okay, well, one reason it's slower is that you might have a scanner, right? A galvanometric scanner to kind of scan the illumination through the volume, and that takes time. But it turns out that even if you have a really fast scanner, fundamentally, the confocal is going to be slower than a wide field microscope for the same signal to noise ratio. And one way of appreciating that is, again, to kind of revisit our pixel grid, right? So now again, you have some n by n pixel grid that builds up the image, but now you're building up the image serially in time, right? And so that means that in any instant in time, your illumination spot is illuminating, in this case, let's say just 1 pixel. And as you move that illumination spot through the sample, then you can build up an image. But again, you're kind of building up the image pixel by pixel. And so what this means is that suppose you take the same amount of time to acquire your image, t. Now, because you're only illuminating 1 pixel at a time, the dwell time per pixel is the time t divided by the number of pixels, right? N squared for an image of n by n pixels. And so that means that if you put in the same amount of power as in our wide field microscope, now each pixel is only giving us a fraction of the amount of signal as the wide field microscope, right? We cut our signal rate by this factor of the number of pixels, n squared. And so what that means is that for the same overall time t it takes to acquire the overall image, our per pixel signal to noise ratio has just been cut by the number of pixels. And so one way of compensating for this issue, right, is to try and, if we look at this equation, try and amplify the power you put in. And you can do that up to a point, but the problem is that because this is a fluorescence imaging system, eventually, if you put in too much power, you saturate the excited state and you don't get any more fluorescence out. And so this is kind of a fundamental limiting factor in a point scanning confocal microscope, that if you increase the power too much, you eventually also increase photo damage, photo bleaching, and saturation. So, no matter how you slice and dice it, compared to a wide field microscope, the confocal microscope gives you fundamentally less signal per unit time. And that's worth appreciating. This just has to do with the way in which you collect data in a serial fashion in a confocal microscope. So I've given you these kinds of two extremes, the wide field microscope and the confocal microscope. And the way I think about light sheet fluorescence microscopy is in some ways getting the best of both worlds. So imagine now bringing in your illumination in a thin plane that, in an ideal case, only illuminates pixels in the focal plane. You could then detect this fluorescence perpendicular to the illumination direction. And now if you imagine what this looks like in our kind of cartoon diagram, this can be very fast because you only have to scan, in this example, in 1 dimension, right, to build up the volume. So it has the same advantage as a wide field microscope in the sense you only scan in 1 dimension because you're illuminating and detecting from all pixels in that plane. But it has one of the advantages of a confocal microscope, namely optical sectioning, because to the extent the light sheet is thin, you can minimize out of focus bleaching and damage. So really the sort of defining characteristic is you're matching your illumination volume to the detection volume. That's kind of the defining characteristic of a light sheet fluorescence microscope. And I haven't said anything yet about how we do this, but hopefully hopefully you can appreciate that sort of where it fits in just on the basis of these simple toy arguments. So I've made this kind of counting argument to sort of suggest to you that a wide field is more efficient than a confocal, and that, in this kind of diagrammatic form, the light sheet is more efficient than either the wide field or the confocal. And so I just want to pause and ask, are there any questions at this point? Does everybody sort of get what I'm saying here so far?
So I have a question about the penetration of the laser, because the longer wavelengths have more penetration than the shortest. So it would also depend on the type of dye or fluorescent protein that you have, and the thickness of the sample of the penetration, because in the entering of the laser, you're going to have better resolution than that in the end. No?
Yes, thank you for that question. And in fact, I will talk about that, Karina. Thank you. Yes, so just like any other form of optical microscopy, if you have very densely scattering tissue, as you say, you will have this problem, right? Especially at low wavelengths where the light is scattered a lot. And this can be evident in light sheet microscopy, because unlike a 2-photon microscope, right? I mean, the entire plane is sort of getting illuminated. And so you can get these kind of scattering effects in scattering tissue. And I'll come back to that. Thank you. Anything else at this point? Okay, let's move on, and there'll be more opportunities to ask questions. So please don't be shy if something comes up. Please ask me. So okay, I just gave you a very bird's eye view of light sheet microscopy. Now I want to delve into some details. So this slide is just giving you an abridged history of light sheet microscopy. And the first thing to realize is that these ideas are not new. So in fact, the first light sheet microscopes, the first recorded ones anyway, go back more than 100 years. Some Germans used light sheets to improve contrast when imaging not fluorescent specimens, but colloidal gold. And one of them actually won a Nobel Prize 20 years later for these discoveries. But then, as far as I can tell, nothing happened for like 90 years. And the next paper where somebody could really sort of rightfully say used a light sheet microscope was at University of Washington in Seattle, where Francis Spellman and his colleagues used what they called the OPFOS to look at cleared guinea pig cochlea. And this paper was sort of forgotten, but really was kind of forward thinking. Then a decade later, some researchers at Scripps used light sheets to look at bacteria in seawater. And I would say it wasn't until Ernst Stelzer at the EMBL realized that this method could be useful for developmental biology. He used a term called selective plane illumination microscopy, or SPIM. And this was, I think, where many others started to get into this game. So there were a few more seminal papers a couple of years later in 2008, again from Ernst Stelzer, but also from Tim Holey at Washington University. Again, folks that just realized that this could be very valuable for doing high-speed imaging or imaging of developmental biology. And then since 2008, there have been more and more people getting into the game, including companies. But I just wanted to give you some sense of the history here, because this is really a case, I think, of an old idea being rediscovered by others in the field, and then more and more people getting into the game. So what actually goes into light sheet microscopy? Well, one of the first things you need is a way of exciting the sample. And in your modern light sheet fluorescence microscope, or LSFM, you typically use lasers. So there's nothing fancy here. You would use often invisible lasers. You can also use 2-photon lasers, and then you need ways of combining the lasers. You might use a fast shutter, like an acousto-optic tunable filter. You would use dichroic mirrors to combine the lasers. Again, there's nothing new here compared to your confocal microscope. Of course, one thing that's sort of unique is making a light sheet. So how do you make a light sheet? Well, there are many ways of doing this as well, but I just want to talk about two of the most common ways. One method is to use what's called a cylindrical lens, right? So if you think about your conventional two-dimensional lens with two surfaces, you would focus a laser, let's say, to a diffraction-limited spot. Well, in a cylindrical lens, you just have one of those axes in one dimension. You don't have any curvature. And in the other dimension, you do. And so what this causes is kind of a light sheet, right? If you look at that illumination profile after the cylindrical lens in the vicinity of the focus from the right direction, it's as if you don't have any focusing. And so this looks like a light sheet. And this method is one of the older methods, but it's still used today. And the advantage is that it's simple. You just put in this optic after a laser beam and you can get a light sheet. One of the disadvantages of this way of doing it is that you have some beam non-uniformity at the edges of the illumination. And so there are ways of dealing with this, but at least sort of from the get-go after the cylindrical lens, this is one drawback, is this kind of beam non-uniformity. Another method of doing it that is also in very common use today is to take sort of a low numerical aperture beam, kind of like a pencil beam, and just scan it at high speed to make a pseudo light sheet. And the advantage of this method is that if you can scan that beam really fast, say compared to the exposure time of the camera, you can get very uniform light sheets. One of the disadvantages, you need an extra scanner, like typically a galvanometric mirror, let's say, to scan the beam. And so just like the point scanning confocal is fundamentally slower than a Widefield microscope, in this way of doing it, if you're scanning a beam to make a light sheet, then this necessarily implies a higher intensity. So for really high-speed imaging, you could run into this problem with fluorescence saturation. But for most imaging at sort of moderate or even pretty fast frame rates, this isn't an issue. And so this is a popular way of generating a light sheet, to just scan a low NA beam at high speed. Then you need basically plumbing, right? What I consider to be optical plumbing, just ways of relaying a beam from one location in your microscope to another. And so there are systems of lenses that you use to relay the focus of one beam to another location, which could be the back focal plane of the microscope objective, or it could be the focus of the objective. And you might have scanning optics like galvanometric mirrors, beam splitters, shutters, and so on. Nothing about this is fundamental. I just wanted to show it so you get some sense of what is there underneath the hood. But again, nothing here is really that unique to a light sheet microscope, right? You would find these kinds of optics in a confocal laser scanning microscope as well. Now, in most light sheet microscopes, you need perpendicular excitation and detection. Now, for every rule, there is an exception to the rule, which I will come back to. But in most light sheet microscopes, I think it's fair to say that you have one objective that introduces the light sheet, another objective that collects the fluorescence orthogonally. And the reason for this orthogonal relationship between the illumination and the detection is that if you detect in most microscopes along the same axis as the illumination, you have this out of focus light, right? So then you would not get the advantage of a light sheet microscope. You want to detect in this kind of perpendicular geometry. And if you do this with two objectives, this places steric constraints on the lenses in your light sheet microscope, right? Because you need to physically fit those two objectives into a confined space around the sample. And so this means that most light sheet microscopes have lower NA objectives than your point scanning confocal microscope. Depending on the sample you're looking at, the NA generally ranges from 0.3 to 1.1. So that's no problem if you're imaging something at low mag, but at high magnification, this can kind of limit the resolution of your light sheet fluorescence microscope. And there's also a working distance constraint as well, right? So if you need to image a specimen that is far enough away that you don't collide with the objective lenses. Detection. So you typically detect with an area detector, unlike a point scanning confocal microscope. And so you would use a high-speed camera. In the old days, everybody used EMCCD cameras, so-called electron multiplying CCDs. These are the most sensitive detectors because you can beat the read noise of the detector by amplifying the signal before you read out. It's also a more mature technique, and these are more costly detectors. You typically have 1,000 by 1,000 pixels on a side, so a megapixel field of view, 30 to 60 frames per second. As I say, negligible read noise, but you do have multiplicative noise when you amplify the signal before it reads out. And so this means you have relatively low dynamic range with an EMCCD. So it can be hard to image samples that have both very dim signal and very bright signal. And for all of these reasons, this generation of cameras are being replaced by this newer generation of so-called scientific CMOS cameras. These are faster and less expensive. You get more pixels per unit time, typically 5 or 10 megapixels. They can be read out at hundreds of frames per second for the whole field of view. And they have pretty low read noise, getting to the limit, I would say, of like 1 electron or less. Much better dynamic range than the EMCCDs. And it's a less mature technology. So some of the kinks are still being worked out, but I think these are getting better all the time. And competition between the vendors is good for us scientists, right? So you can buy a pretty competitive scientific CMOS from many different camera companies with more or less the same performance. So these are the kinds of detectors that you would use in light sheet fluorescence microscopy. I now want to talk a bit about some of the different implementations of this idea, and just starting with one of the more modern implementations from Ernst Seltzer's lab at the EMBL, the Selective Plane Illumination Microscope. In this science paper in 2004, Ernst Seltzer embedded fish embryos, Medica fish embryos, in agarose cylinders, put that agarose cylinder in a water-filled chamber, and then came in from one side with a light sheet that he made with a cylindrical lens, as we discussed, and then detected perpendicularly with a relatively high NA water dipping objective. And if you look at those images of the fish in a Widefield microscope, the fish is big enough and densely labeled enough that the lack of optical sectioning gives you just a garbage image, as you can see here, right? There's very little contrast in the image. On the other hand, in his selective plane illumination microscope, by translating the sample through the light sheet and acquiring one image for every position of the translation, he could build up this beautifully optically sectioned volume. And another advantage of this specimen holder is that you could rotate the specimen. And so that meant that by acquiring one volume at each angle, you could fuse the data together and mitigate scattering in the so-called multi-view reconstruction. So this microscope, it was published more than a decade ago, right? I mean, almost 20 years ago, but still it kind of embodied, I think, some of the strengths of this technique. Okay, so another early implementation from Tim Holy's lab at Washington University was this so-called objective coupled planar illumination microscope, or OCPI. Again, you made a light sheet with a little cylindrical lens, and then the detection lens was physically coupled with this coupling metal piece. And then to take a volume, you would kind of at high speed jiggle the sheet and the detection objective lens together at high speed while recording on an EMCCD. And Tim Holy is a neuroscientist, so he was interested in imaging calcium dynamics in tissue slice. And this was, I think, the first demonstration of using light sheet microscopy to do that. So at the time, this was very fast, imaging these calcium dynamics at tens of hertz. And this method has kind of been forgotten, but it was still a seminal paper at the time. And again, you can kind of see this perpendicular illumination and detection, and then moving both at the same time. So around the same time, Ernst Stelzer published another a beautiful demonstration. It was, in fact, Philip Keller's work. Philip Keller is another leader in this field. And this geometry was kind of similar. The sample, in this case, a zebrafish embryo, was in a water-filled chamber. But now, instead of using a cylindrical lens, they used this high-speed scanning of a low NA beam, the so-called vertically or digitally scanned laser light sheet fluorescence microscope. And they were able to follow almost all of the nuclei during the first the first day of embryogenesis. And this was really kind of an eye-popping movie, right? I mean, this was the first time that many of us paid attention, because this data set, you couldn't have produced this with a point scanning confocal microscope. There was no detectable photobleaching or phototoxicity. The data were good enough. You could actually track thousands of nuclei. This was really something that couldn't be done without this way of doing it. And so this really caused a lot of other people to pay attention. But you sort of see again the kind of similar geometry, right? Illumination from one axis, detection from another in this orthogonal geometry, and then scanning the sample through this digitally scanned light sheet. So I also, developed some light sheet microscopes. At 2011, I developed what I call the inverted microscope-based SPIM, where we kind of built the optics on an inverted microscope-based system. I was really interested in imaging C. elegans embryos, and still am. And so I imaged nuclei in developing C. elegans embryos. And by putting those embryos on a glass cover slip and filling it with buffer and using dipping objectives, I was able to bring in one light sheet from above and then collect the fluorescence from the orthogonal objective. You could scan the light sheet and move the detection lens in sync using a piezoelectric collar. And we were able to record on the order of, I think, 25,000 volumes without any detectable phototoxicity or photobleaching. We've since improved the images, but I think the point is still made that you could image many, many more volumes than a spinning disk microscope without killing the embryo. The embryo actually hatched after continuous imaging one volume every 2 seconds for many hours. And so again, just another implementation of light sheet microscopy. You're really only limited by your creativity and how you build these microscopes, right? So I just wanted to show a couple of other examples. In this example here, showing a sample in a gel coming in from the side with one lens to make the light sheet, coming in from the orthogonal direction to collect the fluorescence. We talked about the ISPIM. You can use high-low geometry to bring in a light sheet. Researchers have also used AFM cantilevers. The advantage here is that you can bring in the light sheet from a different direction, use the AFM to reflect it, and then you could use a high NA detection lens. So you can do single molecule tracking using this approach. And then you can grow samples on a prism, use the prism to reflect the light sheet into the sample, detect from above. So there are many, many ways of doing this. Of course, so Leica has introduced what they call the vertical turn. This is just a mirror that you can put on a confocal microscope. This mirror reflects a confocal scan beam to produce a so-called virtual light sheet. You can then record the light sheet from either above or the bottom. And then there was another interesting idea where some researchers in France built what they called the single objective SPIM, where they just fabricated little micro mirrors, right? And then these micro mirrors had a metallic reflective surface, so you could then use the same objective to introduce the light sheet from the side. The mirror reflects it, and then you could use a high NA lens to detect the fluorescence. So these are just ways of, again, kind of modifying the geometry so as to introduce the light sheet and then collect at high NA. So in this case, using the same objective to both introduce and collect the fluorescence. Of course, here you need these micro-mirrored samples, right? So you need a microfab lab to produce these little micromirrors. Another interesting implementation is this so-called oblique plane microscope, and this is gaining some popularity now. So here the idea is you again use one objective to make the light sheet, and then you use some fancy optics on the detection side. You actually use more objectives and more optics on the detection side to refocus on the detection side away from the initial objective. And the advantage of this is that you can do your refocusing further away. So the sample can be clear of any kind of optics for doing the focusing. You don't have to perturb the sample at all. You can do all of that on the emission side. And this has been popularized by, the seminal idea was Chris Dunsby. It's been popularized by Elizabeth Hillman's group. And one advantage of this way of doing it is you can do very high-speed imaging of whole organisms. In this case, of a larva, a Drosophila larva crawling on a substrate, right? You can record 10 volumes per second, which is fast enough. You can actually record the animal as it's moving around. One of the initial limitations of this approach is that you sacrifice some NA because you're carving up the NA in order to both introduce the light sheet and detect. That concern has been largely ameliorated by work from Bo Huang and Reto Fioca's lab. So you can get almost the entire NA of a high NA lens here. Not quite the whole thing. You're still a bit worse off than a spinning disk or a point scanner, but much closer. And one other disadvantage of this method, I should just say, is that you need a lot of emission optics. You lose something like more than half the light here on the emission side. Nevertheless, I think this is still a useful implementation of light sheet microscopy, and I would encourage you to read more about it if you're interested. Where is this technique really enabling? So in my opinion, I think one area is in developmental biology, where the sample is really delicate and you want to minimize photo damage or photo bleaching, right? So this is where the advantage of using light sheets over illuminating the whole volume can be enabling. Another advantage could be in nervous system imaging. If you want to scan the whole brain of a zebrafish, right, you can do that. This was shown beautifully by Philip Keller some years ago. You can get whole brain recordings. And then, if you sort of push the spatial resolution, you can do recording of single molecules or organelles, as fast as the frame rate allows for many tens of thousands of volumes without frying it. This is just an example from my own lab, looking at caveoli inside a single tissue culture cell at isotropic resolution. And I think another area where this could be enabling is if you just want to image a large tissue volume very fast. So if you have large volumes of tissue and you want to record from many of them, the point scanning confocal gets good images, but is very slow. So if you could use a light sheet, you could record many voxels at once and speed up the images. So it's feasible to image a whole brain in a couple of minutes instead of a couple of days. And so then you kind of shift the burden from the image acquisition to the data, which I'll talk more about later. Then the data becomes the nightmare instead of acquiring the images. So before I go into some of the problems of light sheet microscopy, are there any questions at this point on sort of the hardware or the implementations? Anybody?
Harry, this is Rahul.
Yes.
I have a question regarding this last modality that you showed in where you have the emission side having these objectives trying to image your plane.
Yes.
You talked about the loss in NA.
Yeah.
So are we talking, what kind of loss in NA are we talking about? Are we losing half of the NA, or how is that? Can you explain that?
Yeah. I mean, it depends on kind of how you, this gets into the details of the refocusing, right? But traditionally, one of the problems has been that it's just very difficult to collect all of the light if you're imaging an oblique plane, right? Because you're kind of having to look at it from the side. And so in the early implementations, you would lose something like half the NA just because you're insisting on imaging orthogonally to it. There was a very clever idea by Bo Huang, which is that you can kind of, in the emission side, effectively kind of squeeze that light cone down to a narrower angle and collect more of the NA. And that has then been recently improved again by Reto Fioca and Andrew York. So now they can get almost all of the NA. They still lose a little bit in one direction, but I would say that it's probably fair to say that they're getting like 80 to 90% of the NA, I think would be fair. you know, and the loss is occurring primarily in one direction. In the other two directions, they're kind of getting most of it. Does that sort of help? I mean, it's useful to kind of, look at those papers. I mean, it's still
I'll look them up.
Yeah, I mean, this you know, I would say that this is you know, for applications where you really want the NA. That's, you know, it's sort of somewhat lower mag applications. Maybe you care a little bit less anyway, right? But if you're imaging single cells, right, and you really want to sort of squeeze out the most from it, I mean, I think there's some engineering that one can do that gets you close, for example, to the work you've done, right? But you know, it's still not the same as the work with the AFM cantilever, Rahul, where in principle you can collect it all, right? I mean, you then have to.
I was trying to compare it to the AFM work that Christoph did in Sunny's lab, so that's why I was.
It's still not quite there. I mean, you're still doing a bit better, right, with the work you did, Rahul, but they're getting close.
Okay, all right. Yeah. Thanks.
Sure. Anything else? Okay, we can move on. So I want to talk about some of the challenges in doing this kind of imaging. So one of the early problems was striping in the images taken by these microscopes. And the reason for that is that if you think about what you're doing, right, you're bringing in a plane of illumination, and if the sample has any kind of absorptive or scattering objects inside it, then you will get shadows. And you can sort of see that in this schematic, and you can see that in this data here in this fish. You get these kind of lines, and that's just because you're coming in with a plane of light, and there might be objects that create shadows. Now, this can be ameliorated by taking your light sheet and scanning at high speed. So by pivoting the light sheet at high speed, you have less chances of generating a shadow in any particular direction. And so by doing this in the so-called multi-directional SPIM or MSPIM, you can get much cleaner images. And this has been, I think, commercialized and licensed by Zeiss. So kind of a simple idea to get around this. But if you don't do it and you're imaging a large specimen, you might get these kinds of line artifacts in your image. Another problem that Karina brought up is scattering, right? So if you're imaging a very dense sample, like a Drosophila embryo would be a good example. If you're imaging with one photon illumination, at the edges of the sample, life is pretty good. As you go further in, the illumination light sheet scatters, and that generates scattered fluorescence. And so you can get these gradients in quality from the exterior of the sample as you go further in, which Karina mentioned, right? So in this case, it's obvious in the Drosophila embryo on the inside, the inside of the embryo is imaged quite a bit worse, with worse quality than the outside. And in this case, I think in a zebrafish embryo, the right side or the left side is quite a bit worse if you image from the light sheet coming in from one side or the other. So there are ways of getting around this as well. One sort of obvious idea is to use longer wavelengths and two-photon excitation. So if you build a two-photon light sheet microscope, like a line scanning two-photon microscope, you get much clearer images in the Drosophila, right? If you compare the same sample in the two-photon and the one-photon, that illumination light has less chance to scatter because of the longer wavelength. And even if it does scatter, you're less likely to drive spurious fluorescence because of the two-photon effect. And so this is one way of doing it. The disadvantage is you have to be wealthy. You need to be able to afford a two-photon laser. It's also harder, can be harder to do multicolor imaging, right, with a two-photon illumination scheme, because you then need to rely on fluorophores that have good cross-section and different emission. So that can be a challenge, but this does work. Another simpler approach is just to excite from multiple directions, right? So if there's a pronounced gradient from one side or the other, you could build a light sheet microscope that comes in from one side or the other side, and then you can use image processing to only take the good part of each image and fuse them together. The disadvantage here on the hardware side is you need more hardware. You need to come in from both sides. And of course, you need more dose, right? I mean, you're introducing more dose for every additional view that you take. And then you need to do some image processing. So these are some drawbacks as well of this approach, but it does also work. Another thing you can do is you can introduce some inspiration from confocal microscopy. So if you're imaging very large specimens like a brain, you can imagine on the emission side, if you build up your light sheet using a line scan, using this low NA beam, if you could descan on the emission side, you could pass the stationary fluorescence through a slit to filter out some of the scattered light, and then you could rescan again before you collect on your detector. And that's exactly what these guys did in this paper in 2007. They built a confocal laser light sheet scanning microscope. And if you compare those images to the conventional light sheet microscope, all of this haze is suppressed. And they achieved this by all of these extra optics on the emission side. One of the nice innovations of the scientific CMOS camera is that the CMOS camera itself reads out line by line. So if you can synchronize your illumination to the detection readout of the camera, you can achieve, sort of without all of these extra optics, this confocality, so-called slit scanning or virtual light sheet scanning microscopy. And there was a nice paper by Kubicek and colleagues where they described this. This has been adopted by many others in the field. The advantage is that you can then remove the scatter in real time. The disadvantage is that you're building the light sheet from a line scan instead of a cylindrical light sheet. So this means higher intensities. You also need more synchronization, right? So there's a little bit more investment in time on the hardware synchronization with this approach. Another problem with light sheet microscopy, or really any single view light sheet microscope, is relatively poor axial resolution, right? So this is because of fundamental physics. With any single detection NA lens, in the case of light sheet microscopy, if you look at specimens in the plane of the light sheet, the detection lens images those with higher resolution because you collect more angles, more resolution in plane than out of plane, where the resolution is somewhat coarser. So in this C. elegans embryo, if you image the planes as they come out, the lateral planes look okay. If you look at that volume from the side, now the resolution is smeared a factor of 2 to 3 worse than in the lateral plane. And at somewhat low NA or moderate NA, this becomes even worse. And so this is a problem, can be a problem, right, if you want to get really high resolution in light sheet microscopy. Another kind of related problem is the diffraction of the illumination. So I showed in this cartoon view the light sheet being this kind of not this uniform plane, right, across the specimen. But this picture is a fantasy because any light sheet will spread diffractively. And so the more accurate picture might be one that looks like this, where you have kind of a beam waist, and then that beam waist spreads as it must because of diffraction. And so this means that there is a coupling between the beam waist and the usable field of view. The tighter you make that focus, the thinner the light sheet is, the more spreading there is, and the more bleaching and damage there is at the periphery of your light sheet. And this explains why most light sheets are relatively thick. So if you're imaging, a really large specimen like a tissue section, this isn't a problem, right? You might be willing to cope with a light sheet that is a few microns in thickness. But if you're imaging a single cell and you really care about getting the highest possible Z resolution, this might be a poor choice, right? Because now you're limited not just by the axial resolution, but you're also generating out of focus light on a scale that is larger than the point spread function in Z. And so it would be nice if you could circumvent this problem, both problems actually. And so there are different ways of doing this. One way of trying to do this is to engineer a thinner light sheet, right? So normally we think of generating light sheets or confocal illumination with a Gaussian beam, which overfills or fills most of the NA of your illumination. And this generates a nice tight focus spot. But in light sheet microscopy, it spreads, right? So you don't get this nice focus spot everywhere through your volume. Well, Eric Betzig and others realized that if instead of using a tight focus, you generate an annular focus, you can get what's called a Bessel beam. And this Bessel beam has a property that it maintains its kind of thin, mostly thin structure for a lot longer propagation distance. So if you scan a Bessel beam through a sample instead of a low NA beam, in principle, with this central beam, you can get really high axial confinement. And so there are these gorgeous images in Betzig's papers of single cells resolved at fairly high axial resolution. And the major advantage is you can get down to sort of 300 nanometer axial resolution in live cells. The disadvantage is that if you look carefully at this illumination structure, you see that there are still rings that illuminate the specimen. So even if you can get rid of those rings in post-processing, they're still there. And you're still bleaching your sample more than a Gaussian beam optic. Now, there are ways of trying to do better here. So if you use an array of Bessel beams or a so-called lattice of Bessel beams, you can do a bit better than if you just scan one Bessel beam. you still do worse than a cylindrical lens, and it's hard to maintain this fine beam structure in thick samples. So you might need adaptive optics to maintain this thin structure in thick specimens. So I would say it's not, in my opinion, this is not a uniform win. But for small samples, it can be valuable. And this, of course, has been commercialized in the so-called lattice light sheet microscope. Another way of getting better axial resolution is actually a much older idea, which is inspired by tomography, right? So if you take your sample, in this case, a pollen grain, or in Steltzer's paper here, if you image that pollen grain from one view, you have this kind of anisotropic resolution. The sample looks distorted. But if you can image that sample from many views, kind of like rotating it and taking multiple volumes, if you combine those views, you start to fuse them in such a way that you can kind of isotropize the image of the pollen grain, the volume of the pollen grain. And so with enough views, if you just average them, you can get a more isotropic reconstruction. So this is another idea, right, which is try and take multiple views of the specimen and then fuse them together. And there's an excellent review of this idea in this thesis by Kurzek, published more than a decade ago. So I was kind of inspired by this approach, and I thought, maybe the optimal way of fusing the views is to use joint deconvolution, and that's exactly what we've done. So we've built a microscope that with just 2 views of the specimen, we acquire 2 views with complementary coverage, if you like, in resolution. So in one view, the lateral resolution is good in the plane of the light sheet. The axial resolution is poor. If you can imagine imaging from the other side with a second objective, now you flip the directions of good and bad resolution. Right now, the view in this Z view is pretty good. It's the lateral view. The view in this X view is poor. And if you can collect both of these views, in principle, all the information is there to get an almost isotropic reconstruction, if you can register and deconvolve those views. And so that's something that we built with in collaboration with ASI some years ago, the so-called dual view selective plane illumination microscope. One objective introduces the light sheet, the second objective collects the fluorescence, then you just do this in reverse. The second objective introduces the light sheet, the first objective collects the fluorescence. Because you can switch these views at high speed, the sample doesn't have to move, you can do this relatively fast, and if you register and deconvolve the data, you can improve the axial resolution to double the resolution of a confocal microscope, and the phototoxicity is much less because you're using light sheet instead of confocal illumination. But you still need to do image registration. So the price you pay here is more post-processing. You take, for example, these 2 views, the view A and the view B. After you rotate one view onto the next, because of instrument imperfections, because these objectives are not quite orthogonal, they don't quite overlay. It's close, but not quite correct. And so you then need to do post-processing to finally align these 2 views. And after you register those views, we call that registration, you can then do joint deconvolution to extract the best information from each of these views. And then you can get an isotropic image of the specimen where the resolution is equally good in this axial view as the lateral view. And how does that second part work? How does that joint deconvolution work? Well, it's using a method called Richardson-Lucy deconvolution, which I'll talk more about in the research part of my talk. But in words, the basic idea is that you use an averaged view as a guess. You take these 2 registered views, you average them. You then take that average and you first deconvolve with the point spread function appropriate for the first view. You take that result of the resulting estimate of the sample, you then deconvolve that with the point spread function appropriate for the second view. And by repeating these procedures over and over again, you can end up with an estimate that is a more accurate estimate of the overarching sample with more isotropic resolution. So if you improve this, if you apply this process iteratively, you can get a better estimate than if you just deconvolve from one side at a time. And this is just a few lines of code, so it's not difficult to implement in a computer. And so this really is helpful. We spend a lot of time trying to segment C. elegans nuclei in embryos. And if you compare the single view images to the dual view images, there's a substantial improvement, well, in XY, but notably in Z as well, right? So it actually becomes reasonable to then try and segment these nuclei in these developing embryos. And this works not just for nuclear structure, but it also works for finer structure like neuronal structure. So in this case, looking at a couple of neurons, in the single view images, they're pretty blurred. In the dual view images, they're much better resolved in Z. And so you can actually segment these neurons as they develop in the C. elegans embryo. So hopefully that sort of concept is kind of clear. One other application where I think light sheet microscopy is really enabling is if you're trying to do imaging of cleared tissue. So with the advances of kind of cleared tissue objectives and better refractive index mounting systems and matching systems, you can now look at, say, a mouse brain, the whole mouse brain, and image it in a light sheet microscope. And if we look at sort of a modern microscope to do this, it embodies kind of many of the features that I've talked about. So in this particular microscope, they have two-sided illumination because the sample is really large. And so the light sheet illumination attenuates as you image from one side to the next. You have special objectives and special refractive index liquid to match the refractive index everywhere. They actually use slit scanning, this confocal slit scanning method I mentioned, to kind of reduce scatter, even though it's nominally cleared. And finally, there is some post-processing that is needed to kind of stitch the whole thing together. So all of that is fine. You can do all of that in your instrument or even in a commercial instrument. The real problem is this multi-terabyte data set. So if you want to image, let's say, a mouse brain, 0.3 cubic centimeters at half a micron cubed voxels, that ends up being 4.8 terabytes. And so the data size itself becomes a real pain to deal with. And that's maybe the last point that I want to make, that, if you image in a confocal microscope over one day, where the confocal microscope is recording continuously, you might get something like, near 100 gigabytes of data. In a selective plane illumination mic with an EMCCD, this is now 5 terabytes. And with a scientific CMOS, well, now this is close to 100 terabytes in one color. So multiply that by the number of views and the number of colors. So the data processing becomes something you really have to think about. How do you store the data? How do you compute upon the data? How do you move it around? Maybe you want to use the cloud. And so there's some nice review papers that have been published that discuss how you might think about dealing with the data. I think with better measurement tools, you just get more data. And so there's a real problem in our field right now of kind of just information overload, where you can buy or build a fancy microscope that gives you much more data than a confocal, but actually getting the meaning from that data is not trivial, right? You kind of swap one problem for another problem, which is extracting the meaning from all of this data. So I guess the moral of the story here is you should just think carefully if it actually makes sense to use one of these fancy microscopes. Can you get the job done with a confocal or an ARIScan microscope? You actually have to use these fancier microscopes. And that's something that you have to think about depending on what biology you're studying. But it really can be a challenge when you have these massive data sets. So finally, I just kind of scratched the surface, right? I mean, I just told you about a few different light sheet microscopes. I think there are many options. You can build your own, you can buy them from all of the main microscopy companies now, or you can buy them from these smaller outfitters like ASI or LaVision. Each of these microscopes has strengths and weaknesses. So anybody who claims that their microscope is like the be-all or end-all is just selling you a snake oil. So just approach this with a sort of a skeptical eye when you go into this. And again, I just scratched the surface. There are these excellent resources that have gone into some depth, more than what I could say here in this talk. And I'm also happy to try and answer questions if you want to email me. I'm happy anytime to sort of discuss this.